So unlike carbohydrates and nucleic acids and proteins, lipids are a very large sort of generic class of molecule. The general definition for a lipid is anything that is organic, so it contains carbon, and large, so it's a big molecule, and it's hydrophobic, right? And so there are lots of molecules out there that actually fit under that category. So we can say that lipids are large. Yes. Oh, my fault. Lipids are large, hydrophobic, organic molecules. Okay? And because of that, there's a lot of different types. So we're just going to go through and talk about the little different types here really quick, and then we're going to go into two of the types in much more detail. Okay? So the first type is something that we call a triglyceride. The triglycerides. And these are fats and oils. Okay? So the main purpose of triglycerides, what do you think? What do fats and oils do for us? Yeah.
Burn.
Burn? What do you mean? Burn. You mean like in cars and stuff? Sure. But like on a biological standpoint, what are the main purposes? Yeah.
Don't they like store energy?
Store energy, yeah. So energy storage. Energy storage. And what else? Yeah.
Fats can be used as insulation.
Yes, insulation. And occasionally oils can be used for waterproofing, but we're going to talk more about a better waterproofer in a minute. Okay? So triglycerides, that's one. Phospholipids. Okay, phospholipids are going to have the structure of a phosphate group that has two fatty acid tails on it. And does anybody know what the main thing that the phospholipid is so super important for? Yeah.
Membranes.
Cell membranes are made out of what are called phospholipid bilayers. Okay. There's another type. So triglycerides are three fatty acid tails that's bonded to a glycerol. Phospholipids are two fatty acid tails that are bonded to a phosphate group. The next group are the steroids. Steroids are made up of 4 fused carbon rings. Okay, so no fatty acids at all in here. 4 fused carbon rings. And this is why I said if we were going to talk about lipids and talk about any monomer that they might have, we might say that the monomer is a fatty acid tail because it shows up in the majority of lipids, except we can't say it's in all of them because steroids don't have fatty acid tails. Okay, they're just 4 fused carbon rings. Does anybody know what the main purpose of steroids are? They do. Steroids are a class of molecule that get refined into hormones or chemical messengers. Okay? The last one are the waxes. And waxes are a single fatty acid chain that is bonded to a long alcohol chain, a long carbon-carbon alcohol chain. What are waxes good for? One main job that they have in nature. Waterproofing. If you ever want something to be totally waterproof, the best way to do that is to cover it in a waxy coating. Okay? And because we're not going to go back and talk about them more, next to steroids here we can say 4 fused carbon rings. And then next to waxes, we can say fatty acid, a long chain fatty acid, plus a long chain alcohol. But we're going to go through and talk about in much more detail the triglycerides and the phospholipids because they are the ones that tend to be most important for the biology that we're going to learn about in this class. Okay, so we're going to do structure of triglycerides. Okay, and they're called triglycerides because they are 3 fatty acid tails that are bonded to a glycerol head. So 3 fatty acid tails bonded to a glycerol head. Okay, so we're going to draw the glycerol head here. We're just going to draw it as a box. Glycerol is a, decently large molecule in itself. Okay, and then coming off this glycerol head we're going to have 3 fatty acid tails, and these fatty acid tails can be very long, like 40 or 50 carbons. But we're going to, for our sake of our sanity, we're just going to draw them with like 6 carbons. But they're generally going to be much, much longer than 6 carbons. Something like that. And then if you wanted to draw hydrogen in each open spot, that's fine too. So we got open spots all around here. When you draw a normal structural diagram like this, it's assumed that there are hydrogens in all these spots, but I wanted to kind of put them in so that we don't forget that they're there. It's going to come into play in a second. We put them here because we know that each of the carbons, they have to be bonded to 4 things because it's got 4 bonding sites. And so these ones on the end, for instance, only have 1 carbon-carbon bond, and therefore they're going to have 3 hydrogens on them. Okay? So, first thing about this. Why is this hydrophobic? What makes something hydrophobic? Yeah.
It's not polar.
It's not polar. Yes. Why is this not polar? Why are these bonds here non-polar? Yeah.
Because the hydrogen is separated evenly.
The hydrogens are separated evenly. Yes, that's one thing. Why are the individual bonds, so that's like for molecular polarity, why are the individual bonds non-polar? It's because carbon and hydrogen are very similar in their electronegativities. It's like 2.2 and 2.4, and so that means that there are going to be non-polar bonds between them, which means that the overall molecule is going to be extremely non-polar, and therefore it's going to be hydrophobic. Yeah.
What is the non-polar?
The fact that the molecule itself is pretty symmetrical, right? And then also the fact that each of the bonds, each of the carbon-hydrogen bonds is a nonpolar bond, nonpolar covalent bond, because carbon and hydrogen have very similar electronegativities. Okay? So, nonpolar, that's one of the things that we can know about this. The next thing that we can know about this is that if you look, there are no carbon-carbon double bonds in there at all. In fact, the molecule has all the hydrogens that it could possibly have, right? Because we know that hydrogen is going to go in all the spots where there's no carbon bonded. So this is actually full of hydrogen. It's got more hydrogen in it than, or it's got as much hydrogen in it as it could possibly have. Okay, so we're going to call this. We're going to get a term for it. We're going to call it a saturated fat. Okay, and it's called a saturated fat because, for two reasons. Because it's got as much hydrogen in it as it can. It doesn't have any spots where there could be hydrogen and there's not hydrogen. And the next thing is because it's got all these single bonds. I don't know if you guys are familiar with it. You don't have to draw this at all. But if you imagine two carbons bonded to each other, like this, okay, this bond here is fully rotatable, if that's a word, okay? So this can rotate around like this, right? If this was like a propeller or something like that, then it could spin on that bond, right? A single bond does that. But if it's a double bond, the double bonds come out of the side like this. And so this is locked in place. Double bonds are very rigid, whereas single bonds are spinny, right? And so this is going to allow for these tails to sort of orient themselves in a way where they're laying nice and perfectly flat, all right? So that's going to result in the saturated fat not only having tons of hydrogen in it, as much hydrogen as it can, but it's also going to result in it becoming very dense. Okay, so this is densely saturated. So we can say that there's no double bonds in here. We can also say that this is a very densely packed molecule. And as a result of being very densely packed, it's going to be solid at room temperature. Okay, so over here, I've got last periods, not this periods, reading guides, right? They're like this. They've spent a little bit of time. Would you say these are like overly crinkled up? Does anybody look like they threw this in the trash and then uncrinkled it? No, they're not bad. But they spent a little bit of time in backpacks, and so they got a little bit, roughed up a bit. And you can see it takes up a fair amount of space, right? Everybody see that? Yeah? All right. This is a brand new ream of paper that I haven't even taken out of the paper, right? And this is 500 sheets of paper here, okay? Everybody see this 500 sheets of paper, and they're extremely densely packed. Whereas this is no more than 30 sheets of paper, right? But if you had to guess, this takes up like roughly 1/5 of this ream. So if you were going to use the same density, you would guess that there would be roughly 100 pieces of paper in this, but it turns out there's only 30. And these are just a little bit crinkled up. And so this is much less densely packed because there's a little bit of wrinkles in it, and this is very densely packed because there's no wrinkles in it. If I took this out of here and then like shook it up a little bit, I'd never get it back in because the only reason it's staying in here is because it's perfectly flat, okay? You can think of a saturated fat as being perfectly flat, right? Everything is packed in as tightly as it could possibly go. Whereas in an unsaturated fat, has the same glycerol head. Come on. Okay, so this is the glycerol head. And it's got double bonds. So each time there's a double bond, that's going to form a new angle. Because it's rigid, it can't rotate around. 1, 2, 3, 4, 5, and 6. Okay. 1, 2, 3, 4, 5, and 6. Something like that. Okay, so if we go through and draw the hydrogens then, each of these should be bonded to 3 things, and so this one has 1 hydrogen, 1, 1, 1, 1, and 2. There's 2, 1, 1, 1, 1, and 1. 1, 2, 1, 1, and 3. Okay, so the first thing that you should see is that there's significantly less hydrogens here. Okay, so this is not saturated with hydrogen. It has less than it could. Call this an unsaturated fat. It has carbon-carbon double bonds. Okay, this is the equivalent. This one's really wrinkled up, right? There's a whole lot of double bonds in there. So this one would be extremely less dense than the saturated fat, right? If somebody like crinkled up their paper and then uncrinkled it and turned it in or something like that. But you can see, just like the papers, okay, because they have a little bit of wrinkles in them, they're not going to be able to pack together as tightly. And if they can't pack together as tightly, that's going to result in them being less dense. Okay. And if they're less dense, that means that they are going to be liquid at room temperature. And so we can say that things like animal fats, that if you looked at them at room temperature, they would be solid, right? Those are saturated. Whereas things like oils are unsaturated because they're going to be liquid at room temperature, right? Are we following so far? All right. Which one of these is more energy dense? Which one has more energy per space? Yeah.
Saturated.
Saturated. Not only because you can pack it tighter, but because when we go to get the energy out, it turns out those spots where there's double bonds aren't going to be as useful as a spot where there's a whole bunch of just carbon-hydrogen bonds. Okay? So, you guys ever seen, like, let's say I buy some of those Chips Ahoy soft cookies. You guys ever had the Chips Ahoy soft or like soft batch cookies? They taste not super great. I don't love Chips Ahoy, I'll be up front. But one of the interesting things about them is that if you leave the package open, you would expect them to like get stale and maybe hard or something. They'll always be the same softness, like regardless of how long they've been on the shelf or how long you left the package open, they're always going to be the same. And the reason for that is that they use something that's called partially hydrogenated oil. Okay, and based on the type that you buy, it might be partially hydrogenated soybean oil or partially hydrogenated vegetable oil. The other place that it's really common to find this is in Oreos. Okay, so the center part of the Oreo is made out of partially hydrogenated oil. Okay, so you start with oil that's unsaturated, right? And oil is liquid at room temperature, but you don't want your cookie to be liquid at room temperature. You want it to be like just soft. Okay, so you've got to get it to the perfect amount of softness without, being hard. Okay, so what you want to do is you want to start with an oil that's liquid and you want to hydrogenate it. Okay, and when you hydrogenate it, what does hydrogenate sound like? Hydrogen. So we're adding hydrogens. So we're adding hydrogens through chemical process to make it more saturated. Right? We're taking an unsaturated oil and we're adding hydrogens to it to break the double bonds to make it more saturated so that the density goes up so that we are getting more and more solid at room temperature. We don't want it to be totally solid at room temperature because then it's a hard cookie. We want it to be just a little bit solid at room temperature so that it doesn't like fall apart in our hands, falls apart in our mouth. Okay? So, based on the amount of density that you want, you can hydrogenate it a little bit or you can hydrogenate it a lot. But the point is that if you start with an unsaturated fat, it has to be hydrogenated if you want it to be solid at all. And the more solid you make, or the more hydrogenated it is, the more solid it's going to become because it's going to be more densely packed. Does that make sense? Now, why am I talking about Oreos and soft batch cookies and things like that? Because this is really what nature does too, is it controls the saturation of fats in order to achieve the correct amount of density, right? So it becomes really important when we talk about phospholipids. Phospholipids are next. Phospholipids are a phosphate head and 2 fatty acid tails. Okay. The interesting thing, or the important thing to note about this, is that the phosphate head is going to be hydrophilic because it's got a charge, because phosphate has a -3 charge, right? So it's a hydrophilic head and then these long hydrophobic tails, all right? And as a result of that, when it gets in water, all of the phosphate heads are going to want to point towards the water, and the tails are going to want to point away from the water, because the tails are hydrophobic and the head is hydrophilic. So it's going to form something that is called a phospholipid bilayer. Okay? Phospholipid bilayer has the phosphate groups, that's these red parts. Red parts are phosphate groups. And then the little squigglies are going to be the fatty acid tails. So all the fatty acid tails are going to point towards each other in the middle, like this. And the phosphate heads are going to point out towards the water, because there's probably water out here and water in here. Okay? So this is called a phospholipid bilayer. And the phospholipid bilayer is going to be the thing that makes up the cell membranes, because it is going to be able to keep water in and keep water out. And it does this because it's got all these hydrophobic tails on the inside, and the hydrophobic tails are not going to want to let water pass through them because they're hydrophobic. Okay? So, other stuff has to be able to pass through this, and it has to maintain the correct amount of fluidity, sort of like a soft batch cookie. Right? You don't want it to be too hard, and you don't want it to be too soft. So you want to have just that little Goldilocks moment here where everything is just right. Right? So how does it do that? Well, it changes the amount of saturated and unsaturated fatty acid tails. So if you have a phosphate head here, and it's bonded to 2 fatty acid tails, this one's totally saturated, right? So it'd be really dense, extremely dense, probably too dense. Okay? This one oopsies. Is going to be extremely not dense. It's going to be extremely fluid, right? Because they can't pack together very tightly because it's all these carbon double bonds. Okay? And then you've got the Goldilocks one. And the Goldilocks one might have, you know, mostly single bonds, maybe a double bond here. Maybe a double bond here, but it's mostly just single bonds. And so this one is going to be much more adaptable. It can be in high temperatures, it can be in low temperatures. Whereas this one here, that is all dense, it's all, you know, as solid as it can be, This would be something that you might find in like a hydrothermal vent or something like that. Because as you increase the temperature of something, like let's say, you know, butter's saturated fat, but if you put it in the microwave, it becomes liquid, right? So you can take something that's saturated, and if you increase the temperature enough, it will become fluid. So this might be okay if the organism is living in a hot environment. So all saturated would be for hot environments like hydrothermal vents. This would be all unsaturated. So we would say this would be for cold environments, like, you know, Arctic Ocean or something. And then this is a nice mix of saturated and unsaturated. And this is going to be for like moderate environments. Okay, so just like making cookies, we need to control the amount of saturation in order to control the density and fluidity of the substance. Make sense? Anybody have any questions on this? Okay, I want to talk about a couple things.